This is our first keynote speaker slot at the meeting, and it's my pleasure to introduce Xuemei Chen. Xuemei is the Furuta Chair Professor in the Department of Plant Sciences and Institute of Integrative Genome Biology at UC Riverside. She's also an HHMI investigator and a member of the National Academy of Sciences. Xuemei is an international leader in research on the evolution and function of microRNAs in plants, and she's published some pioneering work on microRNA biogenesis, regulation, and key roles in plant development. And just to give a very brief history, Xuemei, she grew up in Harbin in northern China. She graduated from Beijing University in 1988. And then after working at Caltech and Rutgers, she joined UC Riverside in 2005. So thank you very much, Xuemei, for accepting our invitation, and we look forward to your talk.
Yeah, thank you, Jane, so much for the introduction. Thank you, Zach, Jane, and Sue for putting together such an exciting program. I really enjoyed the meeting so far, and I've learned a lot, and I look forward to the rest of the meeting. Let me share my screen so that I could present. I hope you see it now.
Yep, it looks good.
Great. Yeah. So today I would like to discuss non-canonical RNA caps, such as the NAD, FAD, and DP-CoA caps. I really wanted to raise awareness for the presence of these non-canonical caps in RNA and their potential role in gene expression regulation. Particularly for NAD, we already know that this is a very important molecule in biology. It's a redox agent, and it's found as a cofactor in many enzymes. And now we know that NAD also serves as a RNA cap and can regulate gene expression. And I thought that is also quite fitting that my talk is after the plant microbe interactions session. So in that session, Xin Li mentioned a recent breakthrough in the field, and that is the discovery of tier domain NLRs serving as NADases. So this adds to further the importance of NAD in biology and makes me wonder whether NAD RNAs have any role in plant immunity. So I hope that you will keep NAD RNA capping in mind in your own research endeavor. So in the central dogma, we know that genetic information flows from DNA to RNA to protein. But in the post-genomic era, I think it's perhaps better to represent the central dogma this way. That is, we start from a genome or maybe even genomes and many epigenomes we start with these to get the many epitranscriptomes and also many proteomes. So the gene expression regulation appears to be more complex than we initially thought. And by the epitranscriptome, we refer to RNAs of the same sequence, but with different modifications. For example, RNAs can have different 5' caps, can have different 3' tails, as well as different internal modifications. And all these modifications can impact potentially the translatability, stability, or the subcellular localization of RNA, thus impacting gene expression. And today I'm going to mainly discuss one type of epitranscriptomic modifications, and that is the capping of RNA by non-canonical caps. First, I would like to briefly review the importance of the M7G cap, the canonical cap. So this is a, this is a methylguanosine connected to the first base of the RNA via a first nucleotide of the RNA via a 5' to 5' triphosphate linkage. And this 5' cap is added to the RNA at the co-transcriptional level, but after transcription initiation. So usually after 20 to 25 nucleotides of the RNA have been synthesized by the RNA polymerase, then the capping enzymes deposit the cap onto the RNA. And this cap then influences almost all subsequent processes that the RNA undergoes. For example, in the nucleus, this cap is bound by the CBC, and this cap promotes the splicing and polyadenylation, as well as the nuclear export of the RNA. And in the cytoplasm, the cap is bound by EIF4F to promote translation initiation. And the cap also protects the RNA from degradation. So as we begin to study non-canonical RNA caps, we obviously would wonder what these caps do with respect to these RNA metabolic processes. So as I alluded to, in recent years, some non-canonical RNA caps have emerged, and these caps tend to be cellular metabolites such as NAD, NADH, DP-CoA, and FAD. So these metabolites are actually ATP analogs. And because they're ATP analogs, they can actually be used by RNA polymerase as the initiating nucleotide. So they can be deposited onto the RNA during transcription. So today, I will mainly focus on the RNA NAD cap. And actually, I will be referring to NAD plus and NADH caps together collectively as the RNA NAD cap. And that's because the profiling approaches that I will be talking about cannot effectively distinguish these two types of RNA caps. And then towards the end of my talk, I will also briefly mention our efforts to study FAD and DP-CoA capped RNAs. So first, I would like to give a very brief introduction to the RNA NAD cap. How was it discovered? How is it deposited onto RNA? How is it removed from RNA? And what do we know about the molecular functions of this RNA NAD cap? And I wanted to also say that during this introduction, I will be intermixing knowledge from E. coli, from human cells, and Arabidopsis. So the RNA NAD cap was discovered in 2009 by David Liu's group. So they isolated E. coli total RNA, removed the small molecule fraction, and then subjected the macromolecular fraction to nuclease P1 digestion. So nuclease P1 digests the RNA into nucleotides, and then they subjected the nucleotides to LC-MS analysis. And they were able to detect NAD in RNA. And they further did isotope labeling of RNA and showed that NAD was at the very beginning of RNA. Thus, it was an RNA cap. So using a similar approach, LC-MS approach, we looked into the presence of NAD in Arabidopsis RNA. So we treated Arabidopsis total RNA or mRNA with nucleus P1 or mock treated these RNAs, and then we detected NAD in using LC-MS analysis. And we were able to indeed detect NAD in both total RNA and mRNA. In fact, if you look at the amounts of RNA found in mRNA versus total RNA, it can be concluded that NAD is primarily present in Arabidopsis mRNA. So how is the RNA NAD cap deposited onto RNA? And with respect to this, I think the best known mechanism is actually in E. coli. So in bacteria, it's known that NAD can be deposited onto RNA by RNA polymerase during transcription. So NAD is actually used as the first nucleotide during transcription initiation. And in vitro, eukaryotic Pol2, yeast Pol2 can do this as well. A crystal structure has been solved of a bacterial RNA polymerase together with the DNA template strand in red and the NAD RNA. And from the structure, it can be seen that the NAD moiety in RNA is quite well coordinated by amino acids in the active site of the RNA polymerase. In fact, this NMN moiety of NAD can base pair with the template strand. Because NAD is used as the initiating nucleotide, in bacteria, promoter sequences influence RNA NAD capping efficiency. Also, in human cells, there's some evidence implying that there may be the existence of a post-transcriptional NAD capping mechanism, but this mechanism is as yet unknown. So NAD can also be removed from RNA, and there are at least two types of enzymes that can remove NAD from RNA. One is DXO. So DXO is present in all eukaryotic lineages, such as fungi, plants, and animals. And DXO can remove the entire NAD moiety from RNA, resulting in a 5' monophosphate RNA. A second class of enzymes that denads NAD RNA would be the Nudix family of proteins, and the representative would be E. coli NADC. So this NADC protein can remove the NMN moiety from the NAD RNA, again resulting in a 5' monophosphate RNA. So does Arabidopsis have these enzymes? And the answer is yes. So Arabidopsis has one DXO homolog, and it's called DXO1, and the mutant has very severe phenotypes, as you can see here. In fact, Arabidopsis DXO, as well as DXO from other species, have both two activities. One is the denadding activity. The other is a 5' to 3' exonucleolytic activity that acts on 5' monophosphate RNA. So you can imagine that DXO can probably degrade NAD RNAs by first denadding and then degrading the RNA. In fact, studies from my collaborator E.G. Xia's lab showed that NAD RNA transfected into protoplasts is likely degraded by DXO. So this NAD RNA has very short half-life in wild-type protoplasts, but the half-life is longer in DXO mutant protoplasts. Also in plants, DXO probably degrades NAD RNA, and in the absence of DXO, RNAi can be used as a backup to degrade the RNAs. This was not only found by E.G. Xia's lab, but also found by Joanna Kufo and Brian Gregory's labs. So one major RNAi enzyme is DCL4. So when this mutation was combined with the DXO mutation, the double mutant is seedling lethal, suggesting that RNAi is actually used to degrade NAD RNAs in the absence of DXO. So how about the second family, the Nudix family of denadding enzymes? So we looked for homologs of NADC, and in fact, Arabidopsis has many Nudix domain proteins. And a famous one is actually DCP2. This is the M7G RNA decapping enzyme. So we looked for homologs of NADC, and from our analysis, it seems like NUDT19 is likely to be the ortholog of E. coli NADC. So we performed in vitro biochemical assays to see whether NUDT19 could decap NAD RNA. And I don't think I have time to show you the biochemical data, but we basically found that NUDT19 indeed has the same activity as NUDC, as NADC. So NUDT19 is probably another denadding enzyme in Arabidopsis. So what are the phenotypes of NUDT19 mutants then? In fact, these mutants have already been studied and published, and the NUDT19 loss of function mutant looks just like wild type in terms of development. However, these mutants do seem to differ from wild type in terms of their responses to oxidative stresses. For example, a knockout or a knockdown or knockout mutant, these knockout or knockdown mutants are more resistant to highlight or the oxidizing agent methylviolin. So next, I would like to very briefly discuss the molecular functions of the RNA NAD cap. In E. coli, this cap seems to stabilize RNA. So RNA with this cap has longer half-life as compared to RNA without this cap. But in human cell lines, the results are the opposite. So when these authors from Mike Collision's lab transfected a NAD RNA into this human cell line, they saw that the RNA had shorter half-life, and the half-life became longer in the DXO knockout mutant. So basically, results in this human cell line and in Arabidopsis protoplasts are in agreement. NAD RNAs are destabilized by DXO. In this experiment, they also transfected the cell line with a NAD luciferase RNA, and they were unable to see any luciferase protein being made. So the conclusion was that the NAD cap does not support translation. So that was the introduction to NAD RNAs. And so far, I have not told you what RNAs actually contain the NAD cap. This was only made possible by the development of this NAD capture-seq approach, which I will be talking about next. So this technology was developed in 2015. And basically, this technology first utilizes some reactions to convert NAD RNA to biotin RNA, and the biotinylated RNAs can be purified and sequenced. So the capturing scheme uses these two reactions. The first is an enzymatic reaction using the enzyme ADPRC. So this reaction turns NAD RNA into this alkyne RNA. And the second step is a click chemical reaction. So the alkyne RNA reacts with biotin azide to result in biotin RNA. So using NAD capture-seq, it was found that bacteria, yeast, and human cells have NAD RNAs. And using a similar approach, my lab and Brian Gregory's lab showed that Arabidopsis plants also have NAD RNA. So this was the pipeline that we used for NAD capture-seq with Arabidopsis. I just wanted to use this diagram to show that an important negative control was this sample without ADPRC treatment, without the enzyme. And these, the RNAs that we end up sequencing would represent RNAs that are non-specifically attached to the streptavidin bees. So we subjected inflorescence and seedling RNAs to either ADPRC treatment or not in replicates, and then performed NAD capture-seq. And by comparing the read counts in the ADPRC plus samples and ADPRC minus samples, we were able to identify transcripts that are statistically enriched in the ADPRC plus samples. And these would be the NAD capped RNAs, which I will refer to as NAD RNAs. You can see that we identified thousands of these RNAs, and there was a good overlap between the two types of tissues used. So where are these RNAs from? Which genomes are they made from? As you know, plants have 3 genomes, the nuclear, mitochondrial, and chloroplast genomes. And we found that these NAD RNAs are mainly from the nuclear genome. Some are also from the mitochondrial genome, but intriguingly, none was from the chloroplast genome. And when these RNAs are from the nuclear genome, they can be protein coding genes or non-coding RNAs. And I wanted to mention here that although we did not see any SNO RNAs or snRNAs being NAD capped, they were found to be some were found to be NAD capped by Brian Gregory's lab. I think this is because our capturing seq was did not result in sufficient coverage of NAD SN or SNO RNAs. So by looking at the browser views of these protein coding NAD RNAs, it's very obvious that they do not have introns. So these are the ADPRC plus tracks, and these three are the ADPRC minus tracks. And you can clearly see the removal of these introns. So NAD RNAs are spliced. And we also found that NAD RNAs are polyadenylated. So we first looked into this by RT-PCR on a few NAD RNA gene-producing genes. We did the RT-qPCR after NAD RNA capture. And we did the RT-qPCR with either random primers or oligo-DT primers, and we got similar levels of NAD RNAs. We also looked into this at the genomic scale. So we first isolated poly-A RNA and then subjected poly-A RNA to NAD capture-seq. So here I'm showing you the ADPRC plus minus ratio. This would be a representation of the levels of NAD RNAs. And you can see that there was a very good concordance between poly-A RNA and total RNA, suggesting that NAD RNAs are polyadenylated. Here I wanted to highlight a very innovative sequencing approach developed in my collaborator E.G. Xia's lab at Hong Kong Baptist University. So they modified NAD capture-seq. The first step was the same as NAD capture-seq. So NAD RNA was converted to alkyne RNA, but the second step differed. So instead of using biotin azide, they used a tag RNA covalently linked to the azide for the click chemical reaction. The end result is that the NAD RNA would be covalently linked to the tag RNA. So once they have a library of such RNAs, they subjected this library to Oxford Nanopore single molecule sequencing. So in the single molecule sequencing, you would start from the poly-A at the 3' end of the RNA, and you sequence through your RNA. And if you read into the tag RNA, then you know that the RNA was originally NAD tagged, NAD capped. So because this is a single molecule sequencing, there are actually a few advantages to NAD tag-seq. One is that you can derive the ratio of NAD RNA and M7G RNA from the sequence. So here, if you imagine a mixture of NAD RNA and M7G RNA from any gene, after the capturing scheme, the NAD RNA would be tagged with the tag RNA, but the M7G RNA would not. So if you directly sequence this mixture, then you should be able to estimate the relative abundance of the two types of RNAs. And the estimate from this effort was that NAD RNA is about 5% of M7G RNA. But I think for some NAD RNAs, this is a huge underestimate, as I will talk about later. Another advantage to NAD tag-seq is that you can obtain full-length sequence information, as this is single molecule sequencing. So the NAD RNAs are spliced and polyadenylated. And intriguingly, the NAD RNAs have shorter 5' UTR as compared to M7G RNAs, but they have the same polyadenylation sites. They have the same 3' UTRs. Even though the NAD RNAs appear to be shorter than M7G RNAs, they do contain the entire open reading frames. So this made us wonder whether NAD RNAs can be translated. So we wanted to look into this at the genomic scale, but this question is not that easy to address. So we turned to a question that's more addressable, and that is, are NAD RNAs polysome associated? So we isolated polysomes from Arabidopsis, and then we subjected polysomal RNA to NAD capture-seq. And we also included total RNA isolated from the same samples for comparison. So you can see that we were able to identify many NAD RNAs in the polysomal fraction. In fact, if you look at the levels of NAD RNAs, we actually saw more NAD RNAs in the polysomal fraction as compared to the total RNA fraction. So NAD RNAs are polysome associated. But remember, I told you about the transfection experiment in human cell line, showing that NAD RNA was unable to produce protein. So we looked into whether these polysomal associated NAD RNAs are really associated with translating ribosomes. And we took advantage of this inhibitor of translation, puromycin. So puromycin actually splits translating ATS ribosomes into the ribosomal subunits. And you can see the puromycin treatment worked. We got a reduced amount of polysome and increased amount of non-polysomes. So then we isolated RNA from the non-polysome fraction and the polysome fraction. And then we subjected the RNAs to NAD capture followed by real-time RT-PCR to follow the NAD RNAs from these 3 genes. And you can see that in the mock-treated samples, the NAD RNAs are enriched in the polysomal fraction as compared to the non-polysomal fraction. But after puromycin treatment, this enrichment is either gone or even reversed. We have more NAD RNA now in the non-polysomal fraction. So what this means is that puromycin treatment released NAD RNA from the polysomal fraction and they moved into the non-polysomal fraction. We think that this suggests that NAD RNAs are associated with translating ribosomes, but we still have to look into this further to reconcile the two different results in the human cells versus in Arabidopsis regarding whether NAD RNAs are actually translated. So next, I would like to discuss our efforts to improve upon NAD capture-seq. And I would like to talk about a highly efficient and specific method to profile NAD RNAs, which we call SPARC NAD-seq. So in our efforts to profile NAD RNAs, we realized a problem, and that is if you look at the gene body distribution of the read counts in the ADPRC plus samples, we see this 5' skew in the read count distribution, in the gene body. Whereas in the ADPRC minus samples or in regular RNA-seq, we see a pretty much uniform representation of the reads along the gene body. So we thought about why we would have this 5' skew in the ADPRC plus samples. So we looked into the NAD RNA capturing scheme, and we realized that copper ions present in the click chemical reaction could be causing the problem. So copper ions are known to cause RNA cleavage. So if you imagine RNA cleavage randomly occurring in the body of the RNA, this together with the enrichment for RNA with the 5' biotin would result in a 5' skew in the RNA. So we think that perhaps copper ions were causing the problem. And of course, the copper ion could also lead to reduced efficiency of NAD RNA capture because they were causing RNA degradation. So we decided to see whether we could eliminate this copper, which serves as a catalyst in the click chemical reaction. So this click chemical reaction used in the original NAD capture-seq is copper-catalyzed azide alkyne cycloaddition reaction, which I will refer to as the CoA reaction. So here, the first step was to use ADPRC to convert NAD RNA to this alkyne RNA. And then using this CoA reaction, this alkyne RNA reacts with biotin azide to generate biotinylated RNA. So this was the scheme used in NAD capture-seq. There is actually a click chemical reaction that does not require copper as the catalyst, and this is called strain-promoted azide alkyne cycloaddition reaction, which I will refer to as the SPARC reaction. So in this reaction, a cyclic alkyne is used, and my understanding is that the strain present in the rings allows the reaction to occur in the absence of copper to result in this, this compound. So we thought that maybe we should take advantage of this click chemical reaction to capture NAD RNA. However, this alkyne is so bulky, we thought that maybe it's not a good idea to append NAD RNA to this alkyne. So we wanted to see whether we could use ADPRC to convert NAD RNA to this azide RNA. And then we would add biotin alkyne to this azide RNA to produce biotinylated RNA this way. So in order for this to work, we have to test whether this reaction works. So we first tested the ability of ADPRC to catalyze the reaction between NAD and azide, free NAD and azide. And as a control, this is what was used in the original NAD capturing scheme, where NAD would work with, would react with the alkyne. And indeed, we were able to find products for both reactions, and these products have the mass signatures that indicate that they are the expected products. So it seems like ADPRC can indeed allow NAD to react with azide. So next, we tested the scheme with Arabidopsis RNA. So we would treat Arabidopsis RNA with either the CoA reaction or the Spark reaction to turn them into biotin RNA. And then we run these RNAs in a gel and then detect biotin RNA with streptavidin HRP. So let's first focus on results from total RNA. So if you look at the biotin RNA made with the CoA reaction versus the Spark reaction, you can clearly see that the Spark reaction does not cause RNA degradation, as does the CoA reaction. You also see that there's a background signal from rRNA, even in the absence of ADPRC or the alkyne. And this is most likely due to other modifications in rRNA. So when we used Arabidopsis mRNA to do this, then the negative controls are very clean. As you can see with mRNA, there's a huge signal from the Spark reaction as compared to the CoA reaction, clearly suggesting that the Spark reaction is much more efficient in capturing NAD RNAs. However, this really strong signal also caused some reservation on our part. So we thought that could it be that M7G RNAs are also contributing to the signal? And we went back to the literature and we realized that the original NAD capture-seq technology was developed with bacterial RNA. And it was used actually for eukaryotic RNAs without extensive quality control evaluations to see whether M7G RNAs can also react. So we decided to look into this. So we synthesized NAD RNA and M7G RNA of different sizes in vitro and mixed the two RNAs at different molar ratios and then performed the CoA and the Spark reactions. And here you're seeing the signals of biotin RNAs. As you can see, when the M7G RNA was at a high level, we do see it being converted to biotin RNA. And we suspect that this was also going on with the CoA reaction. It's just that the RNA degradation prevented the identification of the M7G RNA by size. So then we thought that maybe we should try to remove M7G RNAs to prevent them from contaminating the NAD RNA sequencing. And we first looked into whether the anti-M7G antibody that is commercially available to see whether it could help remove M7G RNAs. So we first tried in vitro synthesized RNAs. We used 100 nanogram of NAD RNA and 10 micrograms of M7G RNA, which represent a 1 to 200 molar ratio. We subjected the mixture to no treatment, blank beads treatment, or anti-M7G beads treatment. And then afterwards, we we converted the RNAs to biotin RNA to see the signals of both the NAD RNA and M7G RNA. In the input and also in the blank beads treatment, we saw both being converted to biotin RNA. But after the anti-M7G beads treatment, in the flow-through, we could only see the NAD RNA signal, suggesting that the antibody was effective in removing this large amount of M7G RNA. So then we tried this M7G RNA depletion from Arabidopsis mRNA. And after the depletion, we would digest the RNA with nuclease P1 into nucleotides, and then we followed M7G PPPA as a readout for M7G RNA and NAD as a readout for NAD RNA. And you can see here, upon treatment, upon depletion, 80% of the M7G RNA is gone, whereas NAD RNA is not affected. And we calculated that this 80% reduction should be sufficient to prevent M7G RNAs from contaminating the NAD sequencing. So we combined M7G RNA depletion and the Spark click chemical reaction to develop what we call the Spark NAD-seq. And we compared this with the CoA NAD-seq. And we found that we were able to identify more NAD RNAs, suggesting that it's more sensitive, and higher NAD RNA signal, which also suggests that this is more sensitive, and also more even gene body distribution as compared to the CoA NAD-seq. That shows the 5' skew in the rate distribution. And this you can see better with the gene browser views for individual genes. So the CoA NAD-seq has this 5' skew, but the Spark NAD-seq has even representation along the gene body, which is very similar to RNA-seq. So after removal of M7G RNA contamination, we found gold terms of the top 1,000 NAD RNA producing genes, and they tend to be in the response category, response to various environmental stimuli. And I wanted to mention that the original NAD capture-seq also gave us a lot of response genes. However, we also got photosynthesis as one of the gold categories. And we think that this is likely due to M7G RNA contamination. So these photosynthetic genes are very highly expressed. And we think that the M7G RNAs were perhaps contaminating the NAD-seq. And in the new scheme, in the new technology, we do not see photosynthetic genes as NAD RNA producing genes. So here I wanted to highlight again our collaborators' NAD tag-seq. So they upgraded their NAD tag-seq to NAD tag-seq2 by incorporating the Spark click chemical reaction. And they applied NAD tag-seq2 to E. coli RNA. As E. coli RNA does not have M7G cap, so they did not have to do any M7G RNA depletion in this study. So two take-home messages. One is that if you compare the two phases of E. coli growth, you could see that the amount of NAD RNAs actually changes, suggesting that there's regulation there in terms of RNA NAD capping. And the second message is that for some genes, there is a very high level of NAD RNA. So this is the percentage of NAD RNA in the total transcript for that gene. And you can see that the top one actually has 70% NAD RNA. So this, I think, implies that NAD RNA cap is important in gene expression regulation for at least some genes. Okay, so next, I would like to very briefly discuss our efforts to profile FAD and DP-CoA RNAs. So we know that these metabolites can now serve as RNA caps, and most of the knowledge was actually from LC-MS analysis. So the RNAs from E. coli and human cells were digested into nucleotides, And all these cellular metabolites were found to be present in these RNAs. However, other than the RNA NAD cap, to my knowledge, there have not been any profiling of DP-CoA capped RNAs or FAD capped RNAs. So we don't really know what RNAs contain these metabolic RNA caps. So we decided to try to develop approaches to identify RNAs that have these caps. So there is one pipeline that was already developed to identify RNAs with non-canonical caps. This is called capture-seq. So imagine that you have an RNA that has a non-canonical cap. You can use enzymes that can remove these caps to generate 5' monophosphate RNA. And then the 5' monophosphate RNA can be ligated to an adapter, and then you can do reverse transcription followed by PCR using these handles present in the adapter and the RT primers. So this was actually already done for E. coli, and the problem with this approach is that these enzymes are not so specific to a particular type of RNA cap. So you can basically use this approach to collectively identify RNAs that contain all non-canonical RNA caps. So here I wanted just to show you an example of the specificity of these enzymes that are being used. These are DXO homologs from Schistosaccharomyces pompi. This is called RAY1, and this is mouse DXO. This is Arabidopsis DXO. And NAD-C is the one that already introduced from E. coli that can remove the RNA NAD cap. So here, I won't go into details about these gels. So these are boronate gels that could actually distinguish NAD RNA from, for example, 5' monophosphate RNA by this shift. And if you treat these RNAs with RAY1, for example, you can see that it does not act on M7G RNA. It actually removes the NAD cap from NAD RNA very effectively, but it also acts on FAD RNA. So this is the FAD RNA. After the treatment, the FAD RNA is turned into 5' monophosphate RNA. And it acts also on DP-CoA RNA. So it turns the DP-CoA RNA to 5' monophosphate RNA. And that's the case for NUDC as well as mouse DXO and Arabidopsis DXO. And so what this means is that if you treated these RNAs with, if you treated total RNAs with these enzymes, then you cannot distinguish the caps present in the RNA. So how do we actually specifically profile FAD or DP-CoA RNAs? So we thought about this, and we turned our attention to the Nudix family of proteins. We know at least already that 2 of them are decapping enzymes. So we thought that perhaps more of them are also decapping enzymes, and some may be more specific towards FAD RNA or DP-CoA RNA. So we tested these biochemically, and we found that indeed there are some specificity. For example, let's first look at this, what we call defadding enzyme. So these are NAD RNA, FAD RNA, and DP-CoA RNA treated with the enzyme or not. And you can see that this enzyme does not act on NAD RNA, but it's very active on FAD RNA. And it can also, to some extent, act on DP-CoA RNA. And we found 2 enzymes that are very specific towards DP-CoA. These 2 enzymes do not act on NAD RNA, they do not act on FAD RNA, and they are very efficient on DP-CoA RNA. I'm not showing you the kinetics of the reactions, but we have data on that to show that these enzymes are very specific towards DP-CoA, efficient and specific towards DP-CoA RNA. So once we have these enzymes that have some specificity towards different metabolic RNA caps, then we think that we should be able to devise a profiling approach. So here is just a summary for the activities of these enzymes. So NAD-C is able to remove NAD, FAD, and DP-CoA from RNA. And RAY1 and DXO, they are also able to remove all these caps. So they're not specific. But we have identified a defatting enzyme that's fairly good at FAD RNA, but it also acts on DP-CoA RNA. And we have identified 2 enzymes that are very specific towards DP-CoA RNA. So we thought that what we can do is to first treat total RNA with these enzymes, and then we would turn the DP-CoA RNA into 5' monophosphate RNA, and we can sequence them. And once we have removed the DP-CoA RNAs, then we can treat the remaining RNA with the defatting enzyme, and then we can sequence the FAD RNAs. So this is something that we are developing at the present, so stay tuned. So I think in terms of the future questions, we would like to address whether NAD RNAs are truly translated, and if they are, how are the NAD RNAs recognized by ribosomes? And we are also studying the biological functions of RNA NAD capping and decapping. And we also want to ask whether there are FAD RNAs and DP-CoA RNAs in plants through these profiling approaches that we're trying to develop. So I would like to thank the people who contributed to this work. A current postdoc did a lot of the NAD RNA work that was published together with the bioinformatician Xiaofang Li, a former graduate student, and a current postdoc Hao Hu has done the SPARC NAD-seq development, as well as some of the FAD and DP-CoA sequencing development. And I really want to thank my collaborator, E.G. Xia. We have collaborated over the past 3 to 4 years on NAD RNAs, and the collaboration is really fruitful. These are my funding sources, and I'd like to thank the Metabolomics Core Facility at UCR for the analysis of these metabolites. And thank you for your attention. Yeah, Jane, I hope I didn't go over time.
Don't worry, Xuemei. Keynote is a special talk. Thank you very much. Fascinating mechanisms. We have time. I think we're a little bit over, but there are a few questions. So the first one is from Chin Li. And Chin Li asks, have you tried to fuse GFP gene with the NAD capped RNA to see if the fusion protein can be translated, fused at the C terminus?
Yeah, so Xin, the problem is for any gene that produces NAD RNA, we also see the presence of M7G RNA. So I think it's very hard to do the experiment that you suggested. So even if we made the fusion, we would not be able to tell whether the GFP was being made by the M7G RNA or the NAD RNA. So that is, that is the problem.
Okay. So the second question from Fang Bai. So Fang asks, what percentage of RNA cap is NAD versus FAD or DP-CoA?
So we actually don't know yet whether Arabidopsis RNAs have the FAD or DP-CoA cap. This was shown by LC-MS analysis of viral, bacterial, and mammalian RNAs. So they are found in these RNAs. We are still looking into LC-MS analysis in Arabidopsis RNA, with Arabidopsis RNA. But in terms of the relative abundance, I think this is going to be different for different genes. For example, we suspect that, you may have FAD RNA from one gene, but NAD RNA from another gene. So this we need to do the profiling and also quantification later to know the relative abundance.
Okay, thank you. There's a question about the NAD RNA lifespan, so turnover. Is there anything known about that, the biogenesis versus the degradation?
Yeah, so these transfection experiments in both Arabidopsis protoplasts and human cells show that NAD RNAs are turned over by DXO. So they have shorter half-life than M7G RNA. But if you take away DXO, then the NAD RNA half-life is very similar to M7G RNA. So their half-lives are likely shorter than M7G RNA in a DXO-dependent manner. And also there are additional decapping enzymes such as NUDT19 that I showed, and they probably will also have an impact on the half-lives of NAD RNAs.
And I think so. We can have this final question, Edward Ross. So Edward asks, and says, I think I need to change my paradigm of messenger RNAs now. Yep, agreed. Do you have plans to try to cap specific RNA-seq on stressed versus unstressed plants to see whether these caps play a role in the stress response?
Yeah, very good question. I hope, people in the audience would start to pay attention to these different capped RNAs in your experiments. We cannot do everything. So hopefully, people interested would start to look into this. Yeah, we are also collaborating with people working, in say plant immunity to see whether NAD RNAs are changing, when you apply, say, bacterial or Phytophthora infection. Yeah.
Yeah, very interesting. Maybe I can have a very quick last question, or perhaps on that point. I wondered if the degradation of the NAD RNA, so the removal of the NAD, is that a clean removal, or do you think there are fragments of 5' RNA that are still attached, or degradation products that could then seed the activities of some of these tier domain proteins?
So the decapping enzymes that we know currently, the DXO and the Nudix domain proteins, they either remove the entire NAD moiety or the NMN moiety. So as far as I can tell, after their actions on the RNA would just look like a regular RNA with a 5' monophosphate. Yeah.
Yeah. Thank you very much once again, Xuemei.
Yeah, thank you.